These are the types of bioreactor. So first one is stirrer tank, which we saw in the previous picture. Airlift reactor, packed bed reactor, fluidized bed reactor, photoreactor, and membrane reactor. So this is same what we saw in before. By using an impeller, there are different types of impellers also there. By using this impeller, you will exactly get a good mixing of your nutrients, and also the organism will can able to achieve all the oxygen present inside your nutrient, which is through the spatcher, and it is quite useful for antibiotics and also the enzyme preparation, and also for some of the immobilized enzyme. Then airlift reactor. So airlift reactor is you will have a compartment draft tube. From that, from the end, you will send the sparger inside the inlet compartment, that is tube. The gas will be moving throughout the inlet of which is called as riser portion, and then it will overcome to the sides of the wall, so that the organism is completely mixed, and which is very sensitive to the impeller. So while choosing a microorganism, we should have the we have to understand some of the characteristics and sensitivity of the microorganism, so that we can select which reactor is best for it. Okay, so for mixing purpose, we are using this effective mixing, and also for the stirrer. That is, if it is very sensitive to the stirrer, we are using this airlift reactor. Okay, so for bacterial, animal, plants, and cultures, for fungi, yeast, they are going this for this airlift reactor. Next one is bubble column. Same like Airlift reactor. Thing is that we don't use any draft tube inside it. We will not partition your reactor inside. Instead of it, only this budget will be present. The excess amount of oxygen will be supplied through this bubble column in order to achieve a mixing of your reactor. Okay, this holding up time, everything will be happen. The sparger should send the air bubbles for certain period of time. Okay, from this bubble column reactor, we can say the excellent mass transfer, the liquid and oxygen, the air supply will be very good in this bubble column reactor. The same like what is we are using in our fish tank aeration we used to do, no? So that aerator is similar to. Next one is fluidized bed reactor. So in fluidized bed reactor, we will keep some of the beds, the either the mixture of polymers will be present at the bottom, and they will have a distribution plate at the bottom. From there, they will send the air bubbles. That is, through sparges it will come. So because of the pressure inside it, and also the gas flowing from the downside to upside, what will happen is that along with the air, all the polymers will also move towards, which is a catalyst actually, move towards your solution, and it will start mixing, and it will start distributing all your oxygens from the downside to upside. This is one of the fluidized bed. And it is contrast to the packed bed, whereas that the polymer is loosely packed, whereas here tightly packed. So all the polymers have been tightly packed, and it is settled in only a layer of. Through that layer, if you're sending a sample through your beads, it's a polymer, right? Either it will absorb some of the liquids, or else it will some, if the large molecular weight molecules or water molecules will bypass through the holes present in the polymer. So based upon that, the distribution and also the all suspended particles will be hanged to your polymer, and the reaction will happen through the solids. And after a certain time, the incubation time and all, the reaction time have been left, then it will be removed. We can also say like wastewater treatment, there will be more number of suspended solids will be present, that will be completely removed in the Packedbed column. Then we will move on to our today's class. So photobioreactor. Photobioreactor is from the name itself you can understand. It's an photo. Photo means for the photosynthesis of any plants or algae. We know that algae can grow anywhere in the ponds, lakes, or else in the moisture place. If it is very moist in some of the countries or some of the areas in like Kerala and all, they can see the more number of algae presence will be there in their sand or else in their barrels or something. It will start growing if some of the moisture is present. So the algae requires especially the light source and also the moisture content. So in photobioreactor, we can effectively achieve the production of the algae or some of the plants and some of the microorganism like bacterial. Some of the species requires light source for their production. For that, we will go for this photobioreactor. Okay, you can see it is also same like, and in industries they will use a different layered pattern of photobioreactor because they have to kept outside in order to achieve the growth of your organism. So the organism which requires a light source, then they will go for this photobioreactor. Okay. So they will provide all the specific conditions, all the nutrients which is required for that algae, and then they will also, because of the sunlight, it will start growing inside your reactor, and oxygen supply will also be sent to it, so that the growth can be enormous because usually the algae, fungi, and all will start growing in enormous amount in a rapid divisions of. So that will be very much achieved through this photobioreactor. Okay. So example is that for cyanobacterium and microalgae, we can go for it. Next one is membrane reactor. So membrane reactor is nothing but we will have a reactor, but we have kept some of the membrane inside it um, with the pore size of we can say the micro or else ultra filter pore size. Micro means 0.1 to 5 mm, and for the ultra we can 22,000 Armstrong itself. We'll go for it. What is exactly is that the feed, that is whenever we are inoculating the medium as well as the organism, all the reaction will happen inside it, and it will pass through a membrane from where only the liquids will be overcome through it. All the material our desired products, if we should select, we should select the filter based upon the membrane based upon it. So only that exact pore size can able to enter into it, the microorganism or your products or something will be hold it back. Then after a wash, we can able to achieve it. You can see here the membrane is present inside it. So in that reactor, all the microorganism is utilizing the nutrients present inside it, and it will be held inside your reactor. It cannot pass through it because of its size. Only a few materials, fluids will be moving towards outside, and some of the suspended solids, which is very low in size, will also move from it. Okay. So it is used for the enzymatic reaction and also the primary and secondary metabolites for the plant cells, then also for mammalian cells they go for it. Okay. Thing is that they may require a specific the filtration, the harvesting method will be very sensitive for some of the production units. So for that they will go for this membrane reactor. So exactly all the excess fluids which is not required will be removed in from the reactor itself. Then the organism and desired products will be harvested from the reactor. So in order to minimize the downstream processing, they are going for this membrane reactor. This is what its main use.